Okay. Hello everyone. Welcome to today's Immuseum seminar. So we have a huge treat today, and it's absolutely my honor to welcome today's speaker, Dr. Dan Littman, to our Immuseum seminar series. So before I get started, just a little introduction of Dan. Dan currently is a Campbell professor of molecular immunology, professor of pathology and microbiology in NYU Skirball Institute. Also, he is HHMI investigator. So Dan actually completed his undergraduate study in Princeton, and then also completed his following which he completed his MD PhD training in Washington University, St. Louis. And then after that, Dan started his resident training in pathology in Columbia, and following with his independent research program, actually from UCSF, which I didn't know before. So he actually start from assistant professor all the way to full professor in UCSF in Department of Microbiology and Immunology until 1995. Then he moved to NYU until today. As I mentioned, he's currently a professor of molecular immunology in Department of Pathology and Microbiology, NYU, and also he's investigator of HHMI. So I think a lot of us, including myself, are very familiar with Dan's work. Dan's lab actually dedicated in studying development, signaling, and function of T lymphocytes, and the role of microbiota in shaping immune response in mucosal tissues and in systemic autoimmune diseases. And also his lab studying the machinery of entry and pathogenesis of human immunodeficiency virus. His lab actually really studying a variety of different subjects, which including but not limited to regulation of thymocytes lineage choice, co-receptors in T cell signaling transduction, microbiota immune system interactions, mechanism of HIV and entry of and pathogenesis, and mechanism of Th17-mediated pathogenesis. A lot of recent work from Dan's lab has been aimed at understanding how individual intestinal commensal bacterial species elucidate distinct immune responses. And then the lab has been focusing on identifying microbials and their products that inducing defined T cell response, particular such famous example as SFB-mediated Th17 cell differentiation. And then the goal of this study is to identify intestinal microbials from healthy donors and then IBD patients, which actually correlate with different T cell responses. I think it's truly remarkable that Dan's research really cover a big range of different subjects. But not only that, I think more importantly, a lot of these findings from his lab, really famous examples such as Th17 definition, master transcription regulator, our gamma T4, Th17 cells, SFB, as I just mentioned, now all become the foundation in our textbook. So with all these accomplishments and then numbers of highly cited publication in the field, then receive a really different and a lot of awards and honors, including he's elected a fellow of American Academy of Arts and Sciences. He's also a member of National Academy of Science. He's member of National Academy of Medicine, keynote lecturer, lesson in leadership, AI Distinguished Fellows, etc. The list just keep on going. So, but I think for me, the one of the key important thing impressed me is as a faculty member was in UCSF and currently in NYU, that has been mentored more than 80 different postdoc and students. Many of them become very successful in both academia and industry. And I believe a lot of audience today, myself included, are either directly or indirectly educated by Dan's work during our study and training. So without further ado, it's absolutely my pleasure to have Dan with us today. And we are all very much looking forward to your lecture. Please, the floor is yours.
Thank you so much, Chuan, for this very, very kind introduction. I really appreciate it. I would like to share with you today some things that we're very excited about. It's work that is not yet published, but is provisionally in press and hopefully will be published sometime this summer. It really has to do with what Sean was introducing you to, which is how bacteria, such as in this case, I show you that famous SFB and segmented filamentous bacteria in the background here, how bacteria like this program different kinds of T cells and in the process drive the differentiation of cells in different directions, either at homeostasis or in pathological conditions. So we know that the microbiota that inhabit our gut have largely beneficial functions, promoting immune homeostasis, regulating nutrient acquisition, also promoting signals that basically provide tolerance to various kinds of insults, including protecting from viral infection. Having a good microbiome can also protect from infections with flu and presumably SARS-CoV-2 by basically entraining our immune system to have a particular level of type 1 interferon production that makes it more protective. But when it is disturbed in some way, the microbiota can actually contribute to pathology. It can contribute to autoimmune diseases and inflammatory diseases such as inflammatory bowel disease. It can lead to damage to organs, and it can even influence the response of our immune system against tumors, such as in checkpoint immunotherapy, where we now know from multiple studies that having the right kind of microbiota is really critical for the checkpoint immunotherapy to function. So what, oops, that's a little bit slow there. So, I've depicted these homeostatic functions and the pathogenic functions as shown over here. What we still do not understand is how it is that the microbiota can actually transmit the various signals to achieve these particular aims, both beneficial and pathogenic. So our focus has been on trying to understand the communication of microbiota with cells of the immune system. Also, I won't have time to talk about this today, but also with cells of the nervous system, both the peripheral nervous system and the central nervous system. And then trying to understand the bidirectional signals conveyed between these three different axes in promoting homeostasis and understand what happens when these are dysregulated. So focusing then on the intestine, what we know is that there are multiple types of microbes that make up the microbiota that can inhabit the intestine. I'm only going to focus today on bacteria, but of course we know that there are also viruses, there are fungi, there are protists, there are multiple different kinds of eukaryotic, prokaryotic, and some eukaryotic organisms that inhabit the gut. But the bacteria make up a very large proportion of these, and I'd say we are beginning to understand them better than other kinds of organisms in terms of their interaction with the host. So the bacteria that we all know about are what we can refer to as commensals, which are mutualistic bacteria that provide benefit to the host and derive benefit from the host typically. And then of course, there are pathogenic bacteria. Now the commensals induce homeostatic type of responses, including what we call homeostatic Th17 cells, which actually work to reinforce the intestinal barrier and barriers that other barriers, such as in the skin and the lung. And the pathogenic bacteria typically induce inflammatory types of T cells. They are, of course, invasive organisms largely, and they cause tissue disturbance. But in between, there's a class of bacteria that are not discussed as often, which we are referred to as pathobionts. And these are bacteria that typically have a mutualistic, beneficial, or at least non-harmful relationship with the host. But under certain conditions, be they genetic predisposition or environmental kinds of disturbances, these can become pathogenic. We study one particular kind of pathobiont that has been studied by many labs over the past 3 decades, and that's Helicobacter hepaticus. And what we found a few years ago is that Helicobacter normally induces regulatory T cells, but it also induces inflammatory type Th17 cells that differ really markedly from the homeostatic Th17 cells. They also can make Th1-like cytokines like interferon gamma, and they can cause tissue damage. But the Tregs keep these inflammatory Th17 cells from expanding, but there can be circumstances in which the Tregs can no longer be generated, either due to intrinsic defects, such as the loss of the transcription factor cMAF in Tregs, or extrinsic defects, such as I'll show you in a moment, a number of examples, including the loss of IL-10 signaling. In that case, the loss of Tregs leads to expansion of inflammatory Th17 cells. And we think that this reflects in many ways what happens in human disease, particularly in inflammatory bowel diseases like Crohn's disease, in which we know that there's a disturbance of the microbiota. And a number of laboratories, such as the lab of Jeremiah Faith, have shown that there is a microbiota associated with inflammatory bowel disease that's a pro-inflammatory microbiota, presumably containing a number of pathobionts. So our goal then, experimentally, in order to understand this, is to identify microbial species that are relevant, ideally from human disease, and then identify their biosynthetic pathways that lead to production of metabolites or other bacterial products, and then try to identify the targets of these cells, both the cell targets and the molecular targets in the host of these various bacterial products, and try to understand how these cellular targets then influence the differentiation of the T cells. So that's what I'm going to be talking about today for the rest of my time. So going back then a few years to the work of Mo Xu and Maria Pokrovskii. Mo was a postdoctoral fellow who's now at NIBS in Beijing. And Maria was a graduate student who is now at Calico in California. What they did was to develop a system in which they could track the T cell responses to SFB and Helicobacter hepaticus at the same time. SFB inhabits the ileum, the terminal ileum. Helicobacter inhabits the large intestine. And we know, as I told you, that SFB induces a beneficial kind of Th17 response. And the way we track these responses was by introducing into these mice 2 different kinds of T cell receptor transgenic T cells. One was a T cell that is specific for an SFB peptide, an MHC class 2, another a TCR transgenic T cell that's specific for Helicobacter peptide and MHC class 2. And we could track these different cells based on isotype markers. And at various times after injection, intravenously into either wild type mice or various mutant mice, we can then track the fate of these cells. And as I said, in green here are the SFB specific T cells. These become non-pathogenic Th17 cells that inhabit the small intestine, the ileum. You can see they are almost all ROR gamma T positive. A very small number are these double positive FOXP3 or gamma T positive regulatory T cells, but that's a very minor proportion in this case. However, in the case of Helicobacter, in wild type mice, what you see is that the vast majority of the cells are regulatory T cells. We don't see any of these Th17 cells here. We also see these double negative cells over here. that express BCL6 and CXCR5 and PD1, and those are follicular helper cells. But the key is that these are induced peripheral regulatory T cells that are characterized by expression not only of FOXP3, but also ROR gamma T, and that distinguishes them in large part from the natural or thymic-derived regulatory T cells that are also present in pretty much all different tissues. So this is what's observed when we look in wild-type recipients. If we now look in IL-10 knockout mice, I'm sorry? Hello?
I think there's some background. Go ahead, Dan.
Yeah. Can you hear me now?
Yeah, I can hear you.
If we now look in mice that are deficient for IL-10, and the reason we look in IL-10 deficient mice is because it's been known for many years from work of Alan Scher and Fiona Powry that mice colonized with Helicobacter that lack IL-10 develop an inflammatory colitis. And that inflammatory colitis is marked by Th17-like cells that also resemble Th1 cells. They make both T-bet and ROR gamma T, and I'll show you some of these later, and then make IL-17 also interferon gamma. And you can see that in the IL-10 knockout mice, there are very few of these Treg cells, but instead there are these ROR gamma T only cells. And these cells at the transcriptome level differ really markedly from the non-pathogenic Th17 cells. So our work is really based on this particular find using this system, and I'll tell you about the Helicobacter system that we have now exploited to look in more detail to try to understand how antigen presentation occurs to yield these different kinds of outcomes. So the priming and differentiation of T cells is known to occur in lymph nodes that drain the various tissues. And in this case, we're going to be looking at lymph nodes that drain the large intestine, the so-called C1 mesenteric lymph node in particular. And typically, the paradigm in the field is that dendritic cells that are in the tissues and that can migrate, pick up antigen, and travel through the afferent lymphatics using the chemokine receptor CCR7 attracted to its chemokines CCL19 and 21 to the draining lymph node. And there, these cells go to the paracortical zone where the naive T cells are present. And in this paracortical zone, they now prime the various naive T cells and direct them to become different types of cells. And I've told you about these different kinds of cells that we can observe: the pathogenic Th17, the homeostatic Th17, the induced regulatory T cells, and the follicular helper cells. So the question then that arises is, what is the mechanism for how the antigen-presenting cells can give rise to these different differentiated T cells? So one possibility is that there's a common antigen-presenting cell that is instructed by the microbes to assume the appropriate function and that is required to direct unique T cell differentiation programs. So these are typically thought to be dendritic cells that would be the common cells that would adopt these different programs. Alternatively, there might be distinct antigen-presenting cells, such that microbes would target these distinct pre-existing cells, and each of them would direct a unique T cell differentiation program. So our project really was to set out to try to determine which of these mechanisms was the correct one. So again, the system we use then is to gavage mice with Helicobacter hepaticus, and then a week later, inject the Helicobacter-specific T cells, T cell receptor transgenic T cells, specific for the Helicobacter peptide and IA of B, MHC class 2. And we can label the T cells with CFSC so that we can track them in vivo and track their proliferation. And we can look a few days later at 3 days in the mesenteric lymph node. And if we do that, we can see that these cells have now proliferated. You can see the dilution of CFSC. And they've also upregulated FOXP3. And as you see, they also upregulate ROR gamma T. So these are largely the Tregs that I was showing you. And if we now look yet another week later in the large intestine lamina propria, we see the accumulation of the regulatory T cells and the follicular helper cells. So that's what we see typically in the wild type mouse. And the advantage here is that we can look not only in wild-type mice, but also in the various mutants. But in the wild-type mouse mesenteric lymph node, after colonization with Helicobacter, we can take all the what we consider to be myeloid cells, CD11B, CD11C positive cells, sort them, and perform single cell analysis. In this case, the method we used was CytSeq, which combines single cell RNA sequencing with barcoded antibodies, thus giving us much better resolution of all the different cell types in the lymph node. You can see them labeled over here. You can see that the migratory dendritic cells are largely down here. They are CCR7 positive, and they can be subdivided between CDC2s and CDC1s. And there's been a huge amount of literature in this area over the last decade. And the paradigm in the field is that the migratory CDC2s are the cells that program the differentiation of these peripheral induced regulatory T cells. And part of the reason for this model is that these CD103 positive cells also have the enzymatic machinery for generating retinoic acid. And retinoic acid is known to be critical in the differentiation of regulatory T cells. So this has been the paradigm for the last 15 years, based in large part on work that was done in vitro. And I will show you that it's not quite so simple. So we set out initially to look at the role of the chemokine receptor, CCR7, that I told you is conventionally thought to be required for the migration of the antigen-presenting cells to the draining lymph node. So using a CCR7 conditional mutant mouse, in this case a control, heterozygous, we used the CD11C Cree driver, which is used by many people in the field to look at dendritic cell functions. And then in these mice, we then introduced the naive T cells and looked at CFSE dilution and gene expression and expression of FOXP3 and NOR gamma T. And this is what I showed you earlier. You see the proliferation and upregulation of FOXP3 and ROR gamma T. And this is really a combination of cells that are Treg and follicular helper cells. But when we knock out CCR7, we see one thing that was expected, which is a complete loss of the regulatory T cells. But surprisingly, we still saw very robust priming and proliferation of the T cells. So clearly, there was an antigen-presenting cell bringing Helicobacter antigen to the lymph node. And you can see that these cells express ROR gamma T. And in fact, these are these pathogenic type of Th17 cells that we also observe in the absence of interleukin 10. If we now look about 10 days later in the large intestine, rather than the lymph node, you can see that in the mutants, there's a loss of the Tregs and the accumulation of these Th17 cells. And when we look at the histology, you can see that with the mutants, there's this accumulation of these mononuclear cells. And with time, these animals develop a more severe colitis. And our surprise also, we continue to be surprised because we saw this a very similar phenotype, that is a loss of Tregs and appearance of pathogenic Th17 cells when we use the germline CCR7 knockout. And also when we use CD11C Cree mice driving the loss of MHC class 2. So I'm not going to go into too much more detail here, but regarding these pathogenic Th17 cells. But what we can say is that the antigen-presenting cell that drives the differentiation of these cells is a CD11C negative APC, and it's also CCR7 independent. It's possible that, that there is a cell type that doesn't need to express a particular chemokine receptor because the antigen may migrate directly to the lymph node and then be taken up. Or there could be a antigen-presenting cell, we think likely a monocyte-derived cell that doesn't use CCR7 for its migration. And we're currently looking very hard to try to identify what this cell type might be. But for the regulatory T cell induction, what we can say is that there's a requirement for the CD11C positive cell and expression of MHC class 2, which I didn't show, but also CCR7 in these cells And if we knock either of these out, we lose the Tregs and we get this expansion of the Th17 cells. So our thinking was that this would be a conventional dendritic cell. But we wanted to see whether what might be the actual cells that are targeted by CD11C3. And when we did this, we saw that it's actually quite a breadth of different cell types. Again, using, in this case, CD11C3 to drive a reporter, a Rosa stop TD tomato animal, in which only those cells that express CD11C3 at some point in their ontogeny would now express TD tomato. And we saw the cells that we expected to see, the various kinds of dendritic cells, as well as B cells, which have been described as being CD11C positive. But in addition, we saw a couple of other cell types that I'm going to talk a great deal about. We saw cells that are type 3 innate lymphoid cells that I'll talk more about, as well as a novel population of cells that express the transcription factor AIR, which of course is important in the thymus during selection of the T cell repertoire. It's important for the establishment of tolerance. And these are cells that I'll describe in a few minutes that have been called Janus cells by our collaborators at UC San Francisco. So why did we want to look at these type 3 innate lymphoid cells? Well, the group of Greg Sonnenberg, who I believe is going to speak to you in a couple of weeks in this series, has published a couple of papers over the last 8 years or so, in which they describe the requirement for MHC class 2 expression on type 3 innate lymphoid cells. And in their latest version, they have a model shown over here that the type 3 innate lymphoid cell antigen presentation is required to delete pathogenic Th17 cells. What they saw was that selective deletion of MHC class 2 in the ROR gamma T positive cells using ROR gamma T3 resulted in dysregulated commensal bacteria dependent CD4 T cell responses that promoted spontaneous intestinal inflammation. So this was the model that they promoted most recently. But we decided to look and see whether these presumed type 3 innate lymphoid cells might be important in the regulatory T cell process. And this was actually an incidental observation made by Jimmy Talbot with Maria and Mo a number of years ago, when for other reasons, they were looking in the MHC class 2 knockouts using ROR gamma T3 at differentiation of these Helicobacter-specific T cells. And you can see here in control mice over here, the typical regulatory cells and the follicular helper cells, and they saw exactly the same phenotype upon loss of class 2 in ROR gamma T positive cells, the appearance of these pathogenic Th17 cells and the loss of the regulatory T cells. So that basically led to this model here that the inflammation was not due to the inability to delete effector cells by the ILC3, but rather the inability to drive the differentiation of the regulatory T cells that then allows the inflammatory T cells to expand. I'm sure most of you know what the innate lymphoid cells are, but just to reiterate for you, these are cells that in many ways resemble the programs of T helper cells, but do not have the receptor diversity that T and B cells have. And that's why they're referred to as being innate. But they share the same transcriptional, what people call master regulators that drive their various programs, as well as the cytokines that are made by the various T cells. So for example, a type 1 innate lymphoid cell makes interferon gamma and is dependent on T-bet, just like the Th1 cells make interferon gamma, etc. Now the type 3 cells are the cells that are dependent on ROR gamma T, and in that way resemble the non-pathogenic and pathogenic Th17 cells, and also include a subset that diverges during development from the other ILCs here. That's called a lymphoid tissue inducer cell that expresses CCR6, and these make structures in the intestine called cryptopatches. But all of these cells will be targeted by the ROR gamma T Cre. And it's on the basis of the use of that particular Cre driver line that Sonnenberg and his colleagues concluded that it was the type 3 innate lymphoid cells involved in the process of keeping the Th17 cells in check. So we wanted to look then a little bit more at this. And what we then saw when we looked at the CD11C Cre mice driving the loss of MHC class 2 was that if we now gated on these type 3 innate lymphoid cells, in this case, we gated on these lymphoid tissue inducer cells that are CCR6 positive, but we see that most of these cells now lose the expression of MHC class 2, raising the possibility that all along we were looking at the loss of these genes, not necessarily in dendritic cells where they will be lost, but also in the type 3 innate lymphoid cells. And in fact, when we did these experiments with a number of different target genes, we saw that the CD11C Cre and ROR gamma T Cre mice that were bred to these conditional knockout mice resulted in very similar phenotypes with the loss of the iTreg cells and the expansion of the Th17 cells. So I told you about MHC class 2. We also knocked out H2M using a mouse made by Paul Allen's laboratory. And H2M is required for displacement of the invariant chain during loading of antigenic peptide onto MHC class 2 molecules. So that also led to the loss of Tregs, indicating that the machinery for MHC class 2 presentation is important here. Also, when we knocked out alpha V integrin. Now, alpha V pairs with beta 6 and beta 8 integrins to release TGF beta. I'll talk more about that next. And CCR7, as I have told you about. So what is going on then? We wanted then to go back and do fate mapping with ROR gamma T Cre and compare to what we observed with the dendritic cell gene expression. First of all, coming back to the CD11C Cre mice that I told you about, if we now look at in these fate map mice at expression of MHC class 2, of course you see it on the dendritic cells, but you also see it on these so-called JCs, the Janus cells, and the type 3 innate lymphoid cells. So this is where the Janus cells come in, because they were described in a paper late last year from Jay Gardner's group and Mark Anderson's group at UCSF. The title of their paper was Single Cell Multiomics Defines Tolerogenic Extrathymic Air-Expressing Populations with Unique Homology to Thymic Epithelium. So these cells resemble medullary thymic epithelial cells, the MTECs, but also have features of dendritic cells, and that's why they call them two-faced Janus cells. When we bred mice that were fate mapped for ROR gamma T with TD tomato, and also with green fluorescent protein expressed from a dendritic cell driver that also expresses in these other cell types and gives a very similar phenotype when used as a Cre mouse to the CD11C Cre mouse. What we saw is that the overlap when we gated on both TD tomato and GFP positive cells and sorted these and did site-seq, we saw that the overlap was largely on ILC3 and JC. And this is shown maybe a bit more clearly here, that we can sort the cells into 3 different populations from the lymph node. And you see here mostly the dendritic cells with ZDC GFP, but the dual expression is largely in these air positive cells, Janus cells, and the type 3 innate lymphoid cells. So this left us with the possibility that it would be either or both of these populations that we're looking at, with and without some kind of a dendritic cell population in the induction of the regulatory T cells. And this is where I want to introduce the issue of the integrin that typically is thought to lead to the release of TGF-beta. I told you that we looked at integrin alpha V, and we knocked it out with using ROR gamma T Cre. And in that case, now what we see in the T cells that the Helicobacter-specific T cells, is that they now begin to express both T-bet and interferon gamma. And you can see here that they lose the expression of FOXP3 in the intestine. They still express ROR gamma T and T-bet, as I showed you, and also lose the expression of CCR6, which is not so important for our story here. But we could phenocopy this set of results here by using an antibody made by Dean Shepard's lab that targets integrin beta 8. And you can see when we use this in vivo, we see exactly the same phenotype here, the loss of the Treg and the appearance of these T-bet positive, ROR gamma T positive cells. So what is this integrin? Well, a beautiful paper in Cell a couple of years ago from a couple of groups at structural biologists at UCSF began to shed some light into how this integrin functions. And this alpha V beta 8 heterodimer interacts with latent TGF-beta, as shown over here, basically releasing the TGF-beta, not so that it floats away, but rather that it serves kind of like a lasso and can now recruit the TGF-beta receptor on the same cell that expresses this platform molecule called GARP along with the latent TGF-beta. So this is how the activation is thought to occur. It's known that regulatory T cells can make TGF-beta and can also make GARP. And they also require signaling through the TGF-beta receptor heterodimer. So very likely, whatever cell is involved here, a ROR gamma T positive cell, CD11C positive cell that would be down here, expressing alpha V beta 8 integrin, would be doing this release to activate the differentiation process of the regulatory T cell. So we teamed up with Jay Gardner's group at UCSF to look in more detail, in this case, using single cell RNA sequencing to better define these Janus cells and the type 3 innate lymphoid cells so we could distinguish them. And what they found is that there are 3 different subsets in their clustering of Janus cells, looking now at pooled lymph nodes. And they express, of course, MHC class 2. At least a couple of them express high levels of air. They also express co-stimulatory molecules, CD80, CD86, CCR7, which is critical, and also alpha V and beta 8. But you notice beta 8 is expressed at very low level on the type 3 innate lymphoid cells by comparison. So we collaborated with a group in Lyon in France, Helena Paidasi, who made a reporter mouse in which he knocked in TD tomato into the integrin beta 8 locus. So we were able to gate on cells from the draining lymph node of these animals to look for type 3 innate lymphoid cells and Janus cells. And when we were very careful to exclude any dendritic cells, what we found is that the Janus cells express a relatively high level of integrin beta 8. Not very high, but you can see it here compared to what we see in control mice. And on the y-axis here, sorry, it's not labeled, is the TD tomato expression. But you see that a small proportion of the type 3 innate lymphoid cells also express this TD tomato compared to the background control here. And if we look at proportion of expression, you can see that in JCs, there's a much higher frequency of these cells expressing beta 8 than in type 3 innate lymphoid cells. But if we now look at total numbers in an adult mesenteric lymph node, you see that they're roughly equivalent in total numbers. So we cannot yet state which are the cells that are doing this presentation of alpha V beta 8. It could be both, but we We do not know yet. So the model then that we are left with is that there's a ROR gamma T positive cell that must express MHC class 2, H2M, CCR7, and the integrin alpha V, could be either a Janus cell or a type 3 innate lymphoid cell, and that drives the differentiation of the microbiota-specific Itreg cell. And in the absence of any of these, there is the expansion of these pathogenic Th17 cells. So what we can say is that the current model that I presented at the beginning cannot be a correct one. There cannot just be a conventional dendritic cell driving the differentiation of these Helicobacter-specific peripheral regulatory T cells. One possibility is that there are two different antigen-presenting cells that are required, and we actually favored this model for a long time, such that the DC2s would be doing the priming, and we know that they're terrifically effective at priming naive T cells, whereas either the ILC3 or JC would be also docking with the same cells through the same antigen presentation, but these would release TGF-beta and lead to the differentiation of the Tregs. And because CD11C3 affects the TGF-beta release both in the dendritic cell and the ROR gamma T positive cells, it's very difficult to tell these apart. I can tell you we did a lot of mixed bone marrow chimeras that gave us no evidence that this kind of mechanism was in play. The second possibility is that there's only a ROR gamma T JC ILC3 positive cell involved, and these cells express all of these different molecules, including the CD11C3, and that they both prime the naive T cell and provide the signal, the TGF-beta signal for the induction of the regulatory T cell program. So we took a couple of different approaches to try to distinguish between these mechanisms. One approach was to try to visualize the interaction in the draining lymph node. And this was work done by Ranit Kedmi with Kai Mesa in our lab. Kai is an expert in intravital microscopy. So they looked in mice in which they could distinguish by fluorescent reporters the type 3 innate lymphoid cells or JCs from dendritic cells and from either endogenous T cells or T cells that have been introduced into these mice, the Helicobacter-specific T cells. So looking in Helicobacter colonized mice, then they used these TCR transgenic T cells that had been bred from a mouse that had been bred with Nur77 GFP. Nur77 is a very early activation gene. So the GFP positive cells would be those cells that had been recently primed and activated. And we could also distinguish these cells from endogenous T cells by labeling them with a blue dye before introducing them into the mice. The ILC3 were distinguished in the recipient mice by ROR gamma T GFP, and the dendritic cell by the ZBTB46 CRE fate mapping the ROSA MK2. So the example here would be that the CDC2s would be MK2 positive. The JCs or ILC3 would be both GFP positive and occasionally MK2 positive as well. But we could distinguish them from these. And the activated T cell would be both GFP positive and positive for the blue dye. So this is an example of the kind of thing we can see. We can see the donor-derived T cell that's specific for Helicobacter. If we look at the GFP channel, we can see that these cells have been activated. They've turned on Nur77. This is a GFP positive ROR gamma T positive cell over here. And this is a dendritic cell likely that expresses the ZBTB red marker. So this is a cell that interacts both with the dendritic cell and one of these ROR gamma T positive cells shown over here. And if we now look at cells that are donor-derived, which do or do not turn on the Nur77 GFP, we can actually distinguish these And if we look at the cells that are Nur77 negative, you see very few of them are adjacent to any ROR gamma T positive cells with or without dendritic cells. But if we look at the Nur77 positive cells, we can see that a sizable proportion of these are adjacent to type 3 innate lymphoid cells, or JCs, and giving some more support that these cells are really critical in the induction. But this still does not tell us whether it's a 1 antigen presenting cell or 2 antigen presenting cell model. And the experiment that actually gave us the answer is depicted here. It's a really lovely experiment done by Ranit Kedmi. So what she did was to make bone marrow chimeras in which the irradiated mice were reconstituted with various types of bone marrow. Bone marrow from mice in which the MHC class II was knocked out with CD11C Cree, bone marrow with or without bone marrow from wild type mice, also bone marrow from mice in which we obtained from Terry Laufer at the University of Pennsylvania, in which the MHC class II T Cree. So we call these MHC class II on, and there's a lock-stop locks cassette in the MHC2 IAB allele, so that the gene is not expressed except when Cree is expressed there, and the stop cassette is deleted. And we could then wait for 8 weeks, inject the Helicobacter-specific T cells after colonizing the mice with Helicobacter, and look to see how these cells differentiate. So if you see here with the wild type bone marrow, as expected, you get the Tregs here, and these double negative cells are the follicular helper cells. And with the CD11C Cree knocking out MHC class II, you can see the loss of the Tregs and the appearance of these ROR gamma T positive pathogenic Th17 cells. And these could be complemented by a mixed chimera with wild type bone marrow. But instead of using wild type bone marrow, we used the MHC class II on bone marrow, we saw a really remarkable and very clear result. And that was that we reconstituted only regulatory T cells. So in this case, the MHC class II is expressed only in ROR gamma T positive cells, and we only get Tregs. We do not get follicular helper cells. And that's shown over here with BCL6 on this axis here. You see that we presence of the FOXP3 positive cells over here. We look then in another set of mice reconstituted with bone marrow, but these were not big mixed bone marrow chimeras. If we just reconstitute with this MHC class II on with ROR gamma T Cree, again, we see the same result here. The Tregs and the absence of these follicular helper cells. But what's really interesting here is that we also use CD11C Cree mice to obtain bone marrow and reconstitute. And here we see something somewhat different. We still see the appearance of the Tregs, but here we also see the appearance of the follicular helper cells, indicating that in the CD11C positive populations that are targeted, there is at least one population of antigen presenting cells that can drive the differentiation of the TFH cells. So that then leads us to this model, which is my final slide, in which I think we have answered the question as to how the microbiota target different kinds of target antigen presenting cells for programming the differentiation of different kinds of T cell programs. So that there are multiple antigen presenting cells, each of which in this case can be targeted by Helicobacter hepaticus, but each of which is purposed with driving the differentiation of a different kind of CD4 T cell. A ROR gamma T positive antigen presenting cell, which we cannot yet distinguish between Janus cells and ILC3. Although I'm happy to discuss this, I think it makes a lot of sense that the Janus cells would be involved because they resemble the cells that are tolerizing in the thymus. These cells drive differentiation of Tregs. They require CCR7, we think, for migration to the draining mesenteric lymph node. A different population of antigen-presenting cells, we think likely monocyte-derived cells that are CD11C negative, that do not require CCR7, drive the differentiation of these pathogenic Th17 cells. But these cells are kept in check by the regulatory T cells. And I should point out, in other experiments that I didn't show you here, looking at SFB-specific T cells, we find that those T cells actually require CD11C positive cells and CCR7 positive cells for their differentiation. So very different from these pathogenic Th17 cells. And finally, I've shown you that there's yet a third antigen-presenting cell that's dependent on CCR7 that is required for the induction of the follicular helper cells. I've put in here provisionally migratory dendritic cells, conventional dendritic cells. We don't know that yet for sure. It could even be B lymphocytes for all we know, because they're also targeted by CD11C Cree. And Terry Laufer's lab has published some data showing that antigen presentation, class 2 presentation on B cells may have a role in follicular helper cell differentiation, or at least maintenance. So that's a remaining possibility. So this is where we currently stand. We still do not know what are the microbial products that can lead to differentiation of these particular cells, or to their any kind of functional specification. They may be pre-specified so that they don't need anything in particular, and it's just a hierarchy of T cells that are being induced simultaneously. These are all questions that we need to resolve. We need to try to understand the spatial distribution of these cells during priming in the lymph node, but also in the tissues that they migrate to subsequently. So I will leave you there with this, and just acknowledge the many people who have been involved in this work. What I showed you today is work largely of Ranit Kedmi, a terrific postdoctoral fellow, but Tariq Najjar and Kai Mesa, shown over here, also made very important contributions, with Lena also helping out with some of the computational work. The computational work was done largely in collaboration with people at the New York Genome Center, Yuan Hao in Rahul Satij's lab, and Marla Stokius, who developed the site-seq and helped us out in the early days after that technique was implemented. We also had help from Helena Paidasi and Jay Gardner's group at UCSF with some of the experiments that I showed you. And we also obtained mice from these folks listed over here. And a lot of this work really derived from the original work from Mo and Maria that really set up this entire system. So thank you very much for your attention, and I'm glad to take any questions in the time that we have.